Hi, I'm Maaly. I'm a machine learning scientist at NML, and I will tell you about the machine learning framework I have developed for enriching metagenomic studies with data from open access articles. So before diving into machine learning, I would like to tell you about metagenomics briefly. So what is metagenomics? Metagenomics involves the use of non-culture methods to identify bacteria or microbes in environmental samples. So simply scientists go to different environments and collect water, air, feces, cloud, insect, food, soil, whatever sample they can get from these environments. And then they do DNA extraction, sequencing, and assembly. And then they use the sequenced assembled DNA to identify the microbes in the environmental samples. Then they start to investigate the role of these microbes in a specific environment and the link between these microbes and specific environmental states. Like for instance, some studies found a link between human gut microbiome and diabetes. Others found that some bacteria in the seawater can degrade plastics. Others found bacteria that can produce antibacterial, antiviral, or anti-tumor compounds. However, to reach this kind of metagenomic analysis results, we need a lot of data about the microbiome environment. So what the metagenomic databases such as Magnify are doing is that they hire curators to describe the microbiome samples using a couple of ontologies. As I mentioned, these samples can come from different environments. And you can imagine that there is no ontology that can cover all the environments, all the environment states, all the experimental methods. So if the curators didn't find a term that described this microbiome sample, they tend to use generic terms. And if generic terms are used, this will not serve well the metagenomic analysis. And accordingly, we will not reach really accurate metagenomic analysis results or interpretation. Accordingly, the project I'm working on is aiming for the development of an automated approach that can extract all this data from the publications describing the microbiome samples. So what I've done is that I've developed a machine learning framework that can extract these data from the publication describing the metagenomic studies. So this framework encompassed three components: the literature classification and triage, named entity recognition, and database enrichment. So because I know that most of the biome are kind of biased toward human and marine, and I didn't want really to train models on this kind of biased data. So I was interested at the beginning to cover different types of biome environments. To do that, I needed to train a classifier that can categorize metagenomic papers into three main environments, which are the engineered, the natural environment, and the host-associated environment. Once the classifiers were trained, I applied them on metagenomics papers, categorized this paper, selected a random subset of these papers with evenly distributed biomes, and then used them for the second component, the named entity recognition. For this component, I've defined 16 novel metagenomic instances, curated them, and trained them with BioBERT models. Then I used this named entity recognition models to annotate and extract all the data of metagenomic studies from publications in Europmc. I've annotated around 98K publications, including 1,500 publication links to metagenomic studies in Magnify and 14K publication linked to metagenomic studies in E&E. And you can see here the size of enrichment that has been achieved by this framework in Europmc, in E&E, and in Magnify. The other figures and the tables show actually the performance of the classifier, the biome classifier, and here this table show the high F scores of the name entity recognition models. So I would like to show you now how you can access these annotations in Europmc. So if you go to Europmc, you can go to advanced search, and then you can scroll down to the annotation section where you can select the annotation provider Metagenomics. By this, you can get all the annotations with Metagenomics, all the publications with Metagenomics annotations. And here are a couple of examples to show you the quality of these annotations. So this is a study from the vaginal microbiome, and you can see here that the name entity recognition managed to identify the sample material, the body size, the host, the state of the host, the place of the sampling, the date of the sampling, and the treatment of the host, the DNA extraction kits, the variable regions, the library strategy, the library construction methods, and the sequencing platform. Here are other studies from the ocular microbiome, and you can see that the model managed to identify all the corresponding entities, including, for instance, the state of the host was keratitis and the age of the host. Here is another study coming from the horses, where the sample was a feces, and again the body size was identified, the state of the horses, the age of the horses, the place, and so on. And lastly, I would like actually, given this, the broad range of metadata fields covered by these annotations, so Magnify now is currently including it as enrichment layer. I would like to thank Molecular Connection curators, San Diego Fragoso from Magnify for their biocuration efforts, and Europmc colleagues for their efforts in annotation integration. And I would like to thank you for listening to my presentation. Thank you.